Hello, my name is Mark Foreman, and on behalf of PassageBio, I'd like to thank Alex TLC for the opportunity to introduce you to our company on this very exciting digital community weekend. I'd first like to start by introducing you to PassageBio. We're a genetics medicines company based in Philadelphia and in developing therapeutics for central nervous system disorders, for which there are no or few approved treatment options. We're formed from a research and development collaboration with Dr. James Wilson at the University of Pennsylvania's gene therapy program. At the bottom, I highlight the PassageBio values, and I'd like to highlight two for this audience. One, that we put patients at the center of every decision and action that we make. And two, the goal is to make an impact to transform patients' lives. We're developing therapies for central nervous system disorders across both rare and large patient populations. We have 3 programs that are in the clinic, and I want to specifically highlight our commitment to the leukodystrophies, including GM1 Gangliosidosis and Krabbe disease, both of which are currently enrolling patients in the clinic, and metachromatic leukodystrophy, which is in preclinical development. Today, I'm going to focus on the 2 programs in the clinic, the GM1 Gangliosidosis program, or PBGM1, and the program on Krabbe's disease, or PBKR03. Before I turn to the specific programs, I'd like to introduce you to the method that we use to administer these potential therapies. We administer the drug into the cerebral spinal fluid, which is the fluid that surrounds the brain and the spinal cord. And specifically, we inject the drug. The drug is administered in the cisterna magna, which is the space at the base of the brain. While relatively new for gene therapy, it is a procedure that has been used for decades to administer drugs to patients. One of the questions many people have asked us is why we're using the approach of administering drugs into the cisterna magna. And the answer is relatively simple. One, work by Jim Wilson's group at the University of Pennsylvania have shown that there's improved delivery to the brain and the spinal cord of the gene. In addition, there's the potential for reduced side effects, specifically side effects toxicity associated with liver inflammation. Now, the downside is the requirement that you would need trained personnel to administer the drug, specifically an interventional radiologist or a neurosurgeon. So with this background, I'd like to now turn our discussion to our first potential treatment, which is GM1 Gangliosidosis, or PBGM1. I'd like to make a few important disclosures regarding our program. One, PBGM1 has not been approved for the treatment of GM1. And 2, the safety and efficacy of this treatment has not been established in humans. So I don't need to tell the audience, but about the devastating effects of GM1 gangliosidosis. It is a lysosomal storage disease caused by mutations encoding the gene GLB1 or beta-galactosidase, and affects the cells of the brain and the spinal cord, as well as organs in the periphery, including the heart and the liver. It's characterized by progressive neurological decline in babies and children, and there are no approved therapies that modify the course of the disease. Now, PBGM1 uses a virus to deliver a functional copy of the gene that's defective, or GLB1 or beta-galactosidase. It's targeting the brain and the spinal cord, as well as the liver, heart, and other organs. And the goal of this program is to see if, by providing a normal gene, can we produce enough enzyme that is deficient in the disease to prevent the buildup of toxic material in the brain and other tissues of the body. Now, this is a schematic of the process. So you see in step 1, we take a normal, healthy copy of the beta-galactosidase gene. We insert this into our viral vector. In this case, we use adeno-associated virus, and a lot of the work in gene therapy is done with this virus. And then this virus is injected into the cerebral spinal fluid. Again, as I mentioned earlier, this is the fluid that flows around the brain and the spinal cord. Some of the cells take up the vector and begin to make dysfunctional normal copy of the beta-galactosidase gene, which is then released into the brain tissue and the surrounding fluid. The secreted enzyme can be taken up by other cells throughout the brain and the body, with the goal of slowing or stopping the progression of the disease, again, by preventing the toxic accumulation of toxic materials in the brain. So with colleagues at the University of Pennsylvania, we tested this hypothesis in an animal model of the disease. In animals that have the gene, have no normal copies of the gene, what we call GLB1 knockout, you see on the left this progressive impairment in neurologic function, shown here, right here, or the early or decreased survival of the animals. With introduction of the gene with introduction of the gene, we get improved neurologic function. So the blue lines, they go down, or improved survival, coming close to 100%. And this was shown to be done in a dose-dependent manner, meaning the higher the doses the animals received, the better their neurologic function and the improved survival. So Imagine1, earlier this year, we initiated Imagine1, which is a study to test the effects of PBGM01 in children with infantile GM1. We're testing two different cohorts of children, those with late infantile GM1 and those with early infantile GM1. And in each group, we're going to test two different doses of the virus. We'll start with the lower dose, and if it's well tolerated, we would then go up to the higher dose. And then if that goes well, then we would expand to a larger group of patients to test the long-term safety and efficacy of the drug. The children be followed for 2 years to assess efficacy, and then an additional 3 years for long-term safety follow-up. Now, this study is the first time the drug is being administered to people, so we're assessing very carefully the risks and the safety issue of the treatment and the procedure. With other gene therapy programs, there are known risks with the AAV, of administering AAV, including things like liver inflammation and damage to nerves that connect the spinal cord to the rest of the body, inflammation at the site of injection, and an effect or reduction in the number in the cells or platelets that can involve with clotting. There's also risks associated with the procedure of the drug, including bleeding, discomfort, and the risks associated with anesthesia that's required to administer the drug. So this, to reiterate what I had shown in an earlier slide, we're again studying two different populations, early onset infantile GM1 or late onset infantile GM1. These are in the former, there's children that have symptoms before 6 months of age, and they can range from 4 to 24 months of age. In the late onset, they have to have symptoms between 6 and 18 months of age, and they have to be 6 to 36 months of age to qualify for the study. And they must have evident genetic confirmation that they have a mutation in the gene and low levels of beta-galactosidase activity in the blood. So where are we enrolling the study? Well, we're currently happy to announce that we have 4 sites that are actively enrolling patients. And we intend to enroll patients in 5 different countries in North and South America and Europe. Patients in the majority of these sites, that are not actively recruiting yet, we anticipate that they will either be active by the end of the year or in early 2022. Patients from around the world will be referred to active sites, and PassageBio will facilitate the transportation of patients and their families to a site the closest site that is active near them. So I'm happy to report that in March of this year, the first patient was dosed with PBGM01. This occurred at the Children's Hospital of St. Peter's University in New Brunswick, New Jersey. Later this year, we expect to release preliminary biomarker and safety data from the first 2 subjects, the subjects in this cohort 1 of the study. And we're targeting expanding the trial, assuming the safety and tolerability is established in cohort 1 into cohorts 2 and 3, which include a higher dose for late infantile children, as well as cohort 3, which is early infantile children, the younger children at the lower dose. Now, I'd like to turn my attention to Krabbe program and the potential of our therapeutic for Krabbe disease, PBKR03. Similar to our GM1 program, I want to emphasize that PBKR03 is not approved by regulatory authorities, and the safety and efficacy has not been established in people. As we all know, Krabbe is another leukodystrophy, this time caused by mutations in the gene encoding galactosylceramidase, which causes a loss of myelin in the neurons in the brain and the periphery. Now, myelin is the material that insulates the neurons in our brain and in the rest of our body, in our entire body, and it's critical to the normal functioning of these neurons. And the disease is characterized by progressive neurological decline, including loss of acquired milestones, peripheral neuropathy, seizures, blindness, and deafness. Now, the approach for Krabbe is quite similar to that being used for the GM1 gangliosidosis. But in this case, we're introducing the GAL-C gene, or the galactosylceramidase gene, which again, being inserted into the viral vector, which can then be administered into the CSF. Some of the cells will take it up, express the protein, and that will be secreted and can be taken up by other cells in the brain and in the periphery. So we've tested PBKR03 in both mouse and dog models of Krabbe's disease. And what I'm showing here is data from the dog model. And in the red, here on the left, you can see that animals that lack a functioning copy of the gene have impaired myelination, a higher score is worse, or reduced survival. Whereas in the green, we see that introducing of the gene of the galactosylceramidase gene causes an improvement in the myelination, similar to what the wild type animal in gray, as well as improved survival of the animals. Now, earlier this year, we also initiated GALAX-C, which is a study to test the efficacy and safety of PBKR03 in children with early infantile Krabbe disease. The study design is also quite similar to that being used for GM1 gangliosidosis. The difference is that the populations we're studying is children 4 to 9 months of age, and a second cohort of younger children 1 to 4 months of age. Each cohort will again test 2 doses, and if we establish safety in these children, we'd expand it to a larger cohort to attempt to study the efficacy of the drug. So the study has 3 phases, and this is similar for both studies. There's a screening phase to determine eligibility, and in this program, that requires that children either be pre-symptomatic or symptomatic, but with first symptoms less than 6 months of age. And they must have a documented mutation in the galactosylceramidase gene, as well as biochemical evidence of loss of the function. The children will receive a single dose of PBKR03. And again, they'll be followed for efficacy and safety at 2 years with an additional long-term safety follow-up for 3 years. Now, this is a duplicate of the slide I showed earlier, but it's just a reminder that this is the first time the drug is being administered to people. So again, we're assessing safety very carefully, following both the administration of the virus and the procedure that's required to administer the virus. In this case, we're enrolling patients from 6 different countries in North and South America, as well as Europe. We have 2 sites that are active, but we again hope to have most of these sites active by the end of the year or by early next year. And we're actively working to enroll our first subject. and patients from around the world will be referred to these active sites. And again, PassageBio will help facilitate the transportation of the patients and their families to a site for if they're interested in participation. So in closing, I'd like to thank Alex TLC for the opportunity to introduce you to PassageBio. And most importantly, I'd like to thank the families and patients for their trust and encouragement as we try to develop these therapies that we hope will help the children with these devastating disorders. And with that, I hope you enjoy the rest of the meeting, and thank you for your attention.